Hi, my name is Julia Stone. Today I will present on cryptosporidiosis. I'll cover signs and symptoms of the disease, pathogenicity and virulence factors of the disease-causing organism, pathogenesis of cryptosporidiosis, epidemiology of the disease, and finally, diagnosis, treatment, and prevention of cryptosporidiosis. So how do you know if you have cryptosporidiosis? Watery diarrhea is the most prevalent symptom of cryptosporidiosis. Other notable symptoms include cramps, nausea, vomiting, weight loss, dehydration, and fever. Some people, however, have no symptoms. If symptoms are going to start, it's generally 2 to 10 days post-infection. These are some probable virulence factors that are under investigation for Cryptosporidium. This parasitophorus vacuole formation is, parasitophorus vacuole is like a bubble in the cell that's made of plasma membrane. It protects the Cryptosporidium from the host cell's defense mechanisms. It's hard to study the virulence factors of Cryptosporidium because it's hard to culture them. So this means it's also hard to knock down or knock out some genes, culture those organisms, and see what the effect is on virulence. However, some virulence factors have been investigated. There are 2 proteins that are adherence factors, the mucin-like glycoprotein and the thrombospondin-related adhesive protein. They both mediate adhesion. This CLS, the circumsporozoite-like glycoprotein, which binds to intestinal epithelial cells in humans and bovines. And this glycoprotein 900, which is one of these mucin-like glycoproteins. It's deposited in trails during gliding motility, and it mediates invasion. In 2013, cellular damage and heat shock protein were suspected but not proven virulence factors, and these contingency genes, which have a high rate of recombination and mutation, which allows for a rapid response to changing conditions, protects the crypto further from the host. So how do you get cryptosporidiosis? Through the fecal-oral route, which includes any mechanism for getting fecal matter into one's mouth. So undercooked contaminated food, accidental ingestion due to diaper changes or sexual activity. The zoonotic route is also a prominent route for getting cryptosporidiosis. In 2015, there were several vet students in Pennsylvania who were infected after handling some crypto-positive calves, and they had not properly removed their personal protective equipment and had not used proper hand washing technique in enough time to protect themselves from accidentally ingesting contaminated fecal matter. In general, drinking water is not a source. However, in 1993, there was a famous outbreak in Milwaukee, Wisconsin. This was the impetus for the long-term 2 enhanced surface water treatment rule, which I did some monitoring for over the past several years. And that is a way to see how much Cryptosporidium is in the source water before it goes through a drinking water treatment plant. Swimming in contaminated water, especially in swimming pools, is another route to infection. Cryptosporidium is resistant to chlorine, and the recommendation to stay out of the water for 2 weeks after diarrhea symptoms have passed is going to be really hard to get children to agree to when it's hot and everyone else is playing in the pool. The past several summers, I've seen news items come up in different parts of the country of cryptosporidiosis outbreaks that are traced to swimming pools where someone was swimming too soon after being sick. Here are some beautiful cows. Cows can host Cryptosporidium in their gut and not be sick, but if we if we ingest Cryptosporidium, we generally get sick. Not everyone does. So when that happens, the thick-walled oocyst goes into the human, travels to the gut, where it very happily reproduces, both sexually and asexually. The oocyst releases the sporozoites, and the trophozoite stage of the organism is active, so it can absorb all the nutrients that it needs. This cycle allows the infection to continue within the host and allows some of the oocysts to travel out with fecal matter and potentially reinfect another human. Cryptosporidium is found worldwide in fresh surface water. For example, this top picture is just upstream of a drinking water treatment plant, and there's plenty of space that might be appealing for geese to land, and then they can deposit fecal matter, and with it Cryptosporidium oocysts that can travel in the water and potentially into the drinking water treatment plant. Specific causes of localized outbreaks generally include swimming pools, inadequate drinking water treatment, and animal handling, as we saw with the vet students in Pennsylvania. How do you know if you have cryptosporidiosis? Well, your doctor has to order some specialized testing, so they would need to suspect cryptosporidiosis. Some of these specialized tests include modified acid-fast staining, which this is an example of. The pink-purple circles are the Cryptosporidium oocysts. Immunofluorescence microscopy. This is a Cryptosporidium oocyst under FITC stain. This is that same oocyst with DAPI staining, where you can see the DAPI is making the nuclei glow blue. So there are 4 nuclei there, which means there are probably 4 sporozoites that would be ready to go and continue that cycle of infection if it weren't stuck to a slide and glowing. This is that same oocyst under DIC, and there's a bunch of debris on this slide, which is there to demonstrate that it's not only the organism that's on the slide. Next to this slide is one of the beads that's used for the one of the beads that's for the immunomagnetic separation. That helps separate the organism from the debris, although clearly not all of it. There are several commercially available kits for enzyme immunoassays. They are generally better than the acid-fast stain and better than microscopy. The sensitivities on these kits range from 66% to 100%, and the specificities from 93% to 100%. Additionally, the CDC has validated a real-time PCR TACMAN method for Cryptosporidium parvum and one for Cryptosporidium hominis, but these are mostly used for research, not for diagnosis. In people with healthy immune systems, generally cryptosporidiosis is self-limiting. However, supportive treatments are recommended. That means drinking more water, maybe drinking some electrolyte solution. Nitazoxanide has been approved to treat diarrhea caused by cryptosporidiosis who have a healthy immune system, but not for people who have a compromised immune system. And that lack of approval is just due to a lack of data. People with HIV, with an HIV positive status, are encouraged to consult their healthcare provider. One thing that has been shown to be effective against cryptosporidiosis in people who are HIV positive is antiretroviral treatment, because that allows the immune system to fight off the cryptosporidium. Cryptosporidiosis may not be curable in people with HIV, and if the immune system takes a nosedive, then the symptoms may return. So how do you keep from getting cryptosporidiosis in the first place? Well, if you're taking care of someone who may be infected, consistent hand washing and proper use of personal protective equipment are key. If you are having sexual practices that put you potentially in contact with fecal matter, then using safer sex practices like barriers, for example, dental dams, for potential oral anal contact, and consistent hand washing will be very helpful. Water utilities have a role in prevention of cryptosporidiosis. They filter and disinfect drinking water, and even though there are consistent levels of cryptosporidium in source water, there are very few incidences of treated drinking water being a source of cryptosporidiosis. Chlorinating upstream wastewater treatment effluent is one helpful option, although, as I've said before, cryptosporidium is resistant to chlorine, and people who are working on direct potable reuse of wastewater treatment effluent are very concerned about keeping cryptosporidium out of that loop. Protecting source water is something that we do in Pennsylvania to prevent cryptosporidiosis. So this picture is a best management practice on a dairy farm in Pennsylvania. The fencing funnels the dairy cows through the stream at this protected location so they don't churn up as much dirt. The dirt can carry cryptosporidium particles further than they can get on their own. This picture, these little green spikes are a bunch of trees that are newly planted to stabilize the stream bank. This helps keep the vegetation in place, and the vegetation will slow the stormwater as it comes rolling off the hills, and that will keep cryptosporidium from getting into the river. This is a detention basin filled with dairy cow manure, and you can see the hills behind it, which are fairly steep. In the frozen winter months, having a detention basin like this, which is also a best management practice for dairy farmers, means that the farmers don't have to go up the steep slopes when they're frozen and icy, and there's not a bunch of manure being spread around when it's just going to run right off into the river. They can wait and spread the manure on the land when, like in the spring, when there are plants that are ready to take up those nutrients and keep the crypto right there on the land instead of getting it in the watershed. No questions.